Blast-induced inflammation in the brain is among the most likely mechanisms initiating neurodegenerative processes. The synergistic effects of systemic and cerebral inflammation are essential mechanisms leading to long-term neurological deficits caused by blast. To show the importance of systemic inflammation in the pathobiology of blast-induced neurological deficits, we performed real-time in vivo imaging of myeloperoxidase activity of activated phagocytes in mice exposed to mild-intensity blast during a 1-month post-injury period. Anesthetized mice were mounted in supine position to the animal holder secured inside the driven section of the helium-driven shock tube and exposed to mild-intensity shock wave. The first group of animals did not have any head or body protection. The second group of animals had a rigid, custom-made plexiglass body armor protecting the torso while the head was exposed. And the third group of animals had their heads protected with custom-made plexiglass helmets covering the skull, face, and the neck while the torso remained exposed. The plexiglass torso or head protection prevented any pressure transfer to the chest, abdomen, or head respectively. 10 minutes before imaging, mice were injected with Xenolite ReadyJect inflammation probe, a chemiluminescent reagent in a ready-to-use format that allows for longitudinal tracking of myeloperoxidase level and inflammation status, in vivo at a dose of 200 milligrams per kilogram or 150 microliters per mouse intraperitoneally. Bioluminescence imaging was performed using the IVIS Imaging System 3D series. During the imaging, the animals were anesthetized with a gas mixture of 1% isoflurane, 66% nitrous oxide, and 33% oxygen using the integrated system for gas anesthesia. The duration of imaging was 5 minutes. To validate substrate injection quality, fluorescence images were taken right before bioluminescence imaging using the 745 nanometer excitation and 800 nanometer emission filter set, and an exposure time between 1 and 5 seconds. Photons were quantified using living image software. The representative photographs show the same animal imaged in both dorsal and ventral positions at the given time point after blast exposure. The intensity of bioluminescence was scaled based on the photon counts. The nose cone that can be discerned on the photos is part of the anesthesia device that is the integral part of the IVIS Imaging System 3D series and does not represent a head cover. This mouse does not have any protection, thus the shockwave freely interacted with the entire body, including the head. The distribution of increased bioluminescence shows the myeloperoxidase activity in multiple organs of a representative mouse subjected to whole-body blast exposure, imaged during the 1-month observation period. Increased activity, thus inflammation, was found in the gastrointestinal tract, with clearly outlined transverse and descending colon, then bilaterally in diaphragmal mediastyle parts of the lungs, as well as in the brain. Interestingly, a temporary decrease in inflammation was observed 3 days after blast. Most probably it's due to anti-inflammatory mechanisms later neutralized by inflammatory and/or injury mechanisms. This animal had the rigid plexiglass cover that prevented any direct transfer of the shock wave's kinetic energy to the brain through the skull. Nevertheless, the distribution of the bioluminescent signal, and thus of inflammation, is comparable to that of the animals without any protection, clearly showing inflammation of the gastrointestinal tract, lungs, and brain. It's noteworthy that the cerebral inflammation in this animal with head protection during the latter post-injury period, at 14 and 30 days, is more intensified than that of the previous animal, which had no protection at all. The conclusion was that the head protection did not prevent inflammation and related degenerative processes in the brain. This animal had the rigid plexiglass cover that prevented any direct transfer of the shockwave's kinetic energy to the chest and abdomen. The distribution of the bioluminescent signal shows inflammation in the brain 1 day after exposure. However, the torso protection successfully prevented systemic and cerebral inflammation during the entire 1-month course of post-injury period. The conclusion was that systemic inflammation and multi-organ response to blast play significant roles in the pathobiology of blast-induced traumatic brain injury. When summarized, current clinical and experimental knowledge shows that blast-induced neurotrauma starts with functional impairments such as cognitive deficits, increased blood-brain barrier permeability, diffusion and perfusion deficits in selected brain areas, impaired information processing due to diffuse axonal changes, and failure of energy metabolism. These functional impairments, especially in the case of mild blast traumatic brain injuries, are often temporary and reversible, and a full functional recuperation takes only days or months after injury. Nevertheless, in the case of repeated blast exposures, and/or whenever physical or mental stress acts synergistically with blast exposure, the neuroprotective mechanisms are overwhelmed and the initial reversible functional impairments give rise to irreversible morphological impairments. Those include cascading molecular and biochemical pathways leading to necrotic and apoptotic cell death in the brain, long-term neurological deficits, irreversible diffuse axonal damage, neuroinflammation, and various proteinopathies, among others.